Welcome to this explainer. Today we're doing something really cool. We're shrinking way down to dive deep into the microscopic machinery that literally makes life possible. You know, when we look at a cell under a basic microscope, it often just looks like a tiny balloon filled with fluid. But beneath that surface lies a bustling, incredibly complex metropolis.
So think about this for a second. How on earth does a cell actually move around without bones or muscles? I mean, we rely on our skeleton to stand up, walk, lift things, right? But a microscopic cell, it somehow manages to crawl across surfaces, divide itself perfectly in two, and shuttle materials back and forth, all without a single bone. No way, right? How is that even possible?
Well, the answer lies in something called the cytoskeleton. Now, despite that name, I want you to completely throw out the idea of a rigid, static skeleton like the one inside you and me. Actually, scratch that. It's more like a bustling, dynamic network of protein filaments. It is an internal framework, sure, but it's continuously assembled and disassembled. It provides cells with shape, mechanical strength, transport, and movement. It's literally a shifting, living infrastructure.
So, to understand this microscopic marvel, here's our quick roadmap for today's explainer. 1. The cell's dynamic framework. 2. The 3 cytoskeleton filaments. 3. Motor proteins and transport. 4. Cell movement and division. And finally, number 5, disease, cancer, and medicine. Let's dive in.
Part 1. The cell's dynamic framework, building the cellular metropolis.
Unlike human architecture, where you pour concrete and just leave it there for decades, this internal scaffolding is built and demolished in real time. It's wild. The cytoskeleton is constantly shifting based on exactly what the cell needs at that exact moment. If a cell needs to stretch out, it builds scaffolding right in that direction. If it needs to squeeze through a tiny tight space, it just breaks down those filaments and rebuilds them somewhere else. It is absolutely the ultimate adaptable structure.
Moving on to Part 2. The 3 cytoskeleton filaments, the infrastructure of the cell.
Okay, let's break down these specific jobs. Think of these as the different types of building materials in our cellular city. First up, we've got actin. These are thin filaments, and they are the major players when it comes to cell movement. Next, we have microtubules. These are hollow tubes made of a protein called tubulin, and you can think of them as the major transit highways for intracellular transport. And finally, we have intermediate filaments. These are rope-like fibers made of various proteins. Now, they don't do a whole lot for transport or movement, but man, they are absolute superstars when it comes to providing high mechanical strength.
Let's actually look a bit closer at those intermediate filaments. Like I said, these are basically the steel girders of the cell. Because they provide such incredibly high mechanical stress resistance, they're vital for maintaining the cell's overall shape, supporting the nucleus, and really stabilizing entire tissues. And depending on the type of cell, these girders are made of totally different proteins. You might recognize keratin, that's in your hair and nails, but there's also vimentin, desmin, neurofilaments in your brain cells, and lamins. At the end of the day, they are all about maintaining structural integrity.
Part 3. Motor proteins and transport, the vehicles of the cellular city.
So the cell actually has 2 completely distinct transit systems, which is just a brilliant division of labor when you think about it. On one side, we have actin-based movement. This uses a specific motor protein called myosin, and it's heavily involved in cell crawling and, get this, the contraction of your very own muscles. On the other side, we've got microtubule-based movement. This system uses different motor proteins called kinesin and dynein. This is the system that handles the internal transport of cargo plus the movement of external structures, like cilia and flagella. But what do they both have in common? Their fuel source? Both of these tiny microscopic engines run on ATP energy.
Let's zoom in on that microtubule transport network for a second. Microtubules act a lot like internal train tracks, but they're directional, meaning they have a plus end and a minus end, and this is absolutely crucial for organizing traffic. The motor protein kinesin acts like a little delivery truck, generally transporting cargo toward the plus end. Meanwhile, dynein hauls cargo toward the minus end. And the craziest part, if you look under a really powerful microscope, kinesin literally looks like it has 2 tiny feet walking along the filament. No joke. They drag massive cellular cargo, stuff like vesicles, organelles, protein complexes, right to their target locations. This is extremely important for long-distance transport, especially in things like your neurons, where materials literally have to travel down these super long axons just to keep your nervous system functioning.
Now, over on the actin side of things, myosin operates through this incredibly elegant 4-step mechanical process. First, ATP hydrolysis kicks in to provide the energy.
That energy then causes the myosin protein to undergo a physical shape change. We call that a conformational change. Because of this new shape, the myosin can interact with and physically grab onto the actin filament, and finally, step 4, it flexes, which generates mechanical force. So imagine multiplying this tiny microscopic flexing by billions of myosin proteins all working in perfect unison, and boom, you get the exact force required to contract your bicep.
Part 4. Cell movement and division. The city in motion.
You know, many cells, like your immune cells hunting down an infection or fibroblasts healing a cut, or even just free-living amoebas, they actually move across surfaces using an amazing actin-based crawling process. And it happens in 5 distinct steps. First, rapid actin polymerization builds new filaments right at the front, physically pushing the membrane forward. This creates a protrusion right at the leading edge. Next, the cell uses these adhesion structures to anchor this new front edge to the surface. Then, using those myosin motors we just talked about, the cell body literally contracts, pulling the bulk of the cell forward. Finally, the rear of the cell detaches, letting the whole back end just snap forward. Build, anchor, pull, release. It's literally a microscopic crawl.
And if you look really closely at that leading edge pushing forward, you'll spot 2 specialized structures. First up are lamellipodia. These are broad, kind of sheet-like extensions packed with dense networks of actin. They act sort of like snowplows, just physically shoving the plasma membrane forward. But protruding out of those sheets are filopodia. These are thin, finger-like bundles of actin. They aren't really the brute force of the operation, they're the scouts. Filopodia probe the environment, they sense their surroundings, manage cell adhesion, and provide directional guidance so the cell knows exactly where it needs to crawl.
But beyond just moving around, the cytoskeleton orchestrates what is arguably the most critical event in all of biology, cell division. During mitosis, microtubules assemble to form something called the mitotic spindle. This spindle physically attaches to the cell's chromosomes and yanks the sister chromatids apart, which ensures each new cell gets the exact right DNA. Then, during cytokinesis, an actin-myosin contractile ring, which is made of actin, forms right around the middle of the cell. It acts almost like pulling a microscopic drawstring on a back, constricting the cell membrane until that one single cell pinches perfectly into 2 brand new daughter cells.
Part 5. Disease, cancer, and medicine. When the infrastructure fails. So what happens when this incredible system gets hijacked? Because the cytoskeleton is so incredibly fundamental, when there are defects in its proteins, it causes some seriously devastating macroscopic diseases. For instance, in muscular dystrophies, genetic defects in these structural proteins cause muscle cells to weaken over time simply because they lose their mechanical strength. In ciliopathies, tiny defects in the microtubule structure of cilia actually disrupt fluid movement across cell surfaces, and that leads to severe developmental and organ disorders. And in neurodegenerative diseases, abnormal cytoskeletal organization breaks down that vital intracellular transport we talked about earlier, causing neurons to eventually fail completely.
But perhaps most alarmingly, cancer cells actively hijack the cytoskeleton, the very same dynamic actin systems that allow a helpful immune cell to crawl toward an infection. Yeah, they are weaponized by cancer cells. These cells alter their own cytoskeletal dynamics to enhance their migration, invade healthy surrounding tissue, and eventually metastasize, spreading to entirely different parts of the body. They literally use the cell's own engine for movement against us. It's wild.
But thankfully, understanding this system also gives modern medicine a really powerful way to fight back. Because cancer cells divide so rapidly, certain anti-cancer drugs, like paclitaxel, vincristine, and vinblastine, specifically target microtubule dynamics. By interfering with those microtubules, they disrupt that mitotic spindle we looked at earlier, literally breaking the cancer cell's ability to divide. And in the lab, scientists can actually study these incredible structures using very specific markers. They use phalladin to bind to actin and highlight the cell's shape. They use alpha-tubulin antibodies to illuminate the microtubule highways, and bimintin antibodies to tag the intermediate filaments. This literally allows researchers to see this invisible, glowing city right under a microscope.
Which brings us to our final takeaway. The cytoskeleton is the cell's internal framework and transportation system. It provides structure while generating the forces required for cell movement, intracellular transport, and cell division. So the next time you flex a muscle, or even just think about how a scratch heals, take a second to ask yourself, isn't it absolutely incredible that all of this relies on a bustling, invisible city of shifting protein beams constantly building and tearing itself down just to keep you alive? But that leaves us with a massive question. If cancer can hijack this invisible city to destroy us, and medicine can target it to save us, what other microscopic secrets are hiding in the cytoskeleton just waiting to be discovered?
Thanks for hanging out with me for this explainer, and keep wondering.